Thank you for the nice introduction, Chloe. And I would like to thank everyone for joining our webinar today. I'll lead you to the world of our single-cell RNA sequencing powered by 10x Chromium technology, as well as walking you through the case studies of application to liver diseases. So here in the first part, I'll briefly introduce single-cell RNA sequencing for 10x Genomics. 10x offers the following single-cell services: single-cell gene expression, single-cell immune profiling, single-cell multi-omic ATAC and gene expression, special gene expression, single-cell ATAC, and targeted gene expression. And the Novogene provides the first two types of service, which are single-cell gene expression and single-cell immune profiling. However, we can do starting with single cells all the way through library prep, sequencing, and BI analysis, while 10x only offers library preparation. In addition, you are also welcome to send us pre-made libraries of multi-omic ATAC plus gene expression, single-cell ATAC, and targeted gene expression for sequencing. And in future, Novogene will also develop more service in single-cell RNA sequencing. As I mentioned in the last slide, Novogene provides two solutions in the single-cell level. One is single-cell gene expression. It can be used to identify real cell types among many cells, to understand the tumor heterogeneity, to characterize complex cell populations, and to discover new biomarkers as well. The other solution is single-cell immune profiling. It functions in profiling immune cell proteinuria, determining antigen specificity of T and B cells, characterizing tumor microenvironments, as well as going beyond traditional cytometry. Generally, single-cell RNA sequencing has been applied in many research fields like stem cell biology, urology, and immunology. Now, let's take a look at the single-cell RNA sequencing workflow. After the sample passed the sample QC, the samples were mixed together with reaction reagents, which contain reverse transcription items. In the meantime, gel beads embedded with tens of thousands of different barcodes are formed. And then the samples will be loaded to 10x Chromium controller. Once the samples are loaded, the cells will be separated into the micro droplets, and the enzyme will be added to the cells at the same time. So in here, a gene is formed. Each functional gene contains a single cell, a gel bead of enzyme, and some room temperature reagents. Within each gene vesicle, a single cell is lysed, and the gel bead is dissolved to feed identically barcoded oligonucleotides into the solution. And then the reverse transcription of poly-A mRNA occurs. Then the libraries will be sequenced in our Illumina sequencer, and the data will be sequenced. The data will be analyzed by Cell Ranger and other Novogene in-house softwares. In the second part is about the details of 10x single-cell RNA sequencing at Novogene. Here is the overview of 10x single-cell RNA sequencing project workflow. Clients are responsible for the first part, preparing samples, and the Novogene will take care of the following parts. Once we receive the samples, we will do cryopreserved cell processing for frozen cells. And then we will perform tissue association to tissues. Sample QC is needed to determine the sample's quality. After samples pass the sample QC, library preparation will be initiated, followed by sequencing and analysis. Regarding the starting materials, we can accept frozen cell suspension, also called cryopreserved cells, fresh tissues, and frozen tissues. Single-cell suspension can be obtained from cell culture, dissociated tissues, cell sorting, and other cell isolation methods. We recommend 500,000 to 1 million cells per sample with viability over 70%, and the samples needed to be packed with dry ice. It is necessary to minimize the froze thaw cycles. We highly recommend people to send fresh tissues. If you have difficulty obtaining single-cell suspension or dissociation tissues, you're welcome to send us fresh tissues. The fresh tissues should be kept in max tissue storage solution in 2 to 8 Celsius degree with packed ice, also called blue ice. And the samples can function best in max solution within 48 hours. If you also have difficulty in getting fresh tissues, we also recommend frozen tissues, and the nuclei isolation will be needed for frozen tissues. Please also make sure that you have at least 50 milligram samples. Frozen tissues need to minimize froze thaw cycles and shipping with dry ice. In addition, sometimes we can also accept fresh cells for some special type of cells. So if you are not sure which cell type which shipping method you will use, then please feel free to contact your cells or your TS. Then once we receive the samples, we will perform sample QC. Here we show the 2 workflows for 2 kinds of starting materials. Workflow A is for tissues and workflow B for cells. If the total amount is less than 50 milligrams for tissue samples, we suggest that clients resend samples. Otherwise, the samples will be proceeded at risk. And then tissue dissociation will be applied to get single cells. For starting material in single cell suspension, we recommend people send over 100,000 of cells with viability over 70%. After QC, if the cell number is insufficient, we suggest to resend samples. That cell removal will be performed when viability is low. And if the viability is still low after that cell removal, we recommend sending new samples. Otherwise, we will move forward at risk. As you can see from this picture here, this is the machine used for account cell viability. It's called the Cellometer Auto 2000. So here I would happy to introduce single cell library types. The first type is 10x single cell 3' gene expression library, which is used to study gene expression of different cell populations. 3' gene expression library is constructed from genes generated by 10x chromium controller. Within the gene vesicles, reverse transcription occurs in order to obtain the cDNA from barcoded poly-A mRNA. As you can see from the picture in the left here, the primer with poly-T is used to capture poly-A mRNA. Once the poly-A mRNA is captured, the reverse transcription reaction will happen, and then template switch oligo, also called TSO, will be added to full-length cDNA that is used for downstream cDNA amplification. In this slide, we also have the structure of 3' gene expression library. It contains 10x barcode, which is 16 base pair single cell identifier, UMI, which is 10 base pair unique molecule identifier, and the 8 base pair sample index, which is used for demultiplexing. Next single cell 5' gene expression library is designed for immune profiling. The library construction process is similar with the 3' gene expression library, but it starts from the other end of the poly-A mRNA transcripts. In addition, the TSO is used differently here compared to single cell 3' assay. In the 3' assay, the poly-T sequence is part of the gel bead oligo, with the TSO supplied in the room temperature primer. In the 5' assay, the poly-T is supplied in room temperature primer, and the TSO is part of the gel bead oligo. and the 10x single-cell VDJ gene expression library are mainly used for exploring immune cells, such as B cells and T cells. And the library construction principle is the same as 5' gene expression library. Now we have a general idea of 10x single-cell 3' gene expression library, 5' gene expression library, and a 10x single-cell VDJ gene expression library. In this slide, we summarize the difference between 3' gene expression and 5' gene expression libraries. 3' gene expression library is designed for almost all kinds of cell types, such as human, animal, plants, and even fungus. 5' gene expression library is designed specifically for researches on immune cells for human and mouse. For VDJ library, we offer the following options: VDJ for T cells, VDJ for B cells, VDJ for T cells and B cells, and the 5' gene expression library plus any one of the 3 options above. The platform for sequencing is NovoSeq 6000, PAL and 150, as shown in the pictures in the right here. The recommended sequencing depth is 100,000 reads per cell, and the maximum targeted cell number is 10,000 cells per sample. Therefore, if researchers targeted 5,000 cells per sample, then the output will be 150 gigabase per sample. Novogene offers both full-length sequencing and partial-length sequencing for 10x single-cell sequencing projects. In this slide, we quoted the 10x company recommendations for sequencing depth. 10x suggested a minimum of 20,000 reads per cell for several kits and 50,000 reads cells for single-cell 3' V2 kit. And they also indicated that the sequencing depth will finally depend on 2 factors. The one is sample type, and the other is the experimental questions being addressed. For example, the sequencing depth of 50,000 reads per cell will only reach 30% to 50% saturation. 50% saturation may be sufficient for cluster your subpopulations of interest during analysis. However, if you are interested in identifying as many genes as possible, you may want to reach 90% saturation level. So the sequencing depth of 50,000 reads per cell is definitely insufficient. After knowing this, then we will go back to see the difference of the recommended coverage between Novogene and the 10x. So why we recommend higher depths like 100,000 reads per cell, while 10x recommend a minimum of 20 or 50,000 reads per cell. So 10x recommends significantly less, but this is just like a basic requirement for performing the analysis, like clustering. But the reason why we recommend higher is that we would like to help clients detect out more genes for most samples, especially for gene expression libraries when low sequencing depths cannot reach the expected saturation level. Again, considering that different samples require different sequencing depths, we can adjust in order to meet the research purpose. So here is an example regarding the sequencing depths. The researchers sequenced the samples at both depths of 17,000 reads per cell and the depth of 145,000 reads per cell. And it revealed that there was no difference in number of clustered cell population, but for further analysis, they chose to use high sequencing depth data. Novogene offers 3 pipelines for 10x single-cell sequencing data analysis. The first pipeline is 10x single-cell gene expression analysis as shown in the flowchart. Sequencing data processing and quality control is analyzed by software cell ranger star, identifying of highly variable genes, cell subpopulation identification, and marker gene detection are analyzed by CIRECT. Cell subpopulation identification includes principal component analysis, identifying clusters of cells, and dimensionality reduction and visualization. Enrichment analysis, such as GO enrichment, KEGG pathway enrichment, reactome enrichment, functional annotation, and protein-protein interaction network is analyzed by a combination of software. The second type is 10x single-cell gene expression cell ranger only analysis. The data will be analyzed only by cell ranger, which includes alignment, gene expression quantification, clustering analysis, differential expression analysis, and visualization. As it's shown, all the analysis was analyzed by cell ranger. The last two types of analysis are for 10x single-cell gene expression libraries, and the third type of analysis is 10x immunoprofiling analysis, including summary matrix, alignment, and the distribution of clonal type, which focuses more on immune cells and analyzed by cell ranger. Liver cancer is a worldwide common cause of cancer-related deaths, with a 5-year survival rate of only 18%. Liver cancer is the second most lethal tumor after pancreatic cancer. In this part, I would like to show some case studies in 10x single-cell RNA sequencing applied to liver diseases. These papers were empowered by Novogene. So let's see how Novogene 10x single-cell RNA sequencing helped in people's research aims. The first paper is from Journal of Nanobiotechnology, which was published in June 2021, named the Single-Cell RNA Sequencing Reveals the Mechanisms of Sonodynamic Therapy combined with a RAS inhibitor in the setting of hepatocellular carcinoma. RAS activation is a frequent event in hepatocellular carcinoma, also called HCC. Combining a RAS inhibitor with traditional clinical therapeutics might be hampered by a variety of side effects. Herein, researchers reported on integrating an IR-820 nanocapsule augmented sonodynamic therapy, also called SDT, with the RAS inhibitor, phenosyl thiosalicylic acid. The single-cell RNA sequencing transcriptional profiles of an HCC xenograft following treatment was used to dissect the mechanisms of a combined tumoricidal therapeutic strategy. The research strategy is shown below. In vitro experiment analyzed how different treatments influence the cell apoptosis. In vivo experiment analyzed how different treatments influence the tumor, and then they applied single-cell RNA sequencing to identify tumor cells composition and changes in signaling pathway to key genes. So finally, the mechanisms on the rational combination of nanocapsule augmented SDT and the RAS inhibitor for highly efficient HCC therapy is revealed. From the picture in the left bottom here, we can get a general idea of the whole process. The researchers obtained HCC xenograft from the mouse, and then they got single-cell suspension. After library preparation, the libraries were sequenced in Illumina NovoSeq sequencer. So here in this slide, we have the plots of single-cell sequencing analysis results. Figure A compared the treatment group A and the control group C. Figure B shows that the cells are finally identified as certain groups, and figure C shows the differentially expressed genes. They were all got from our 10x single-cell analysis package. In this slide, the researchers draw the conclusion from the plots that among the cluster between group A and group C, clusters 6, 7, 12, and 13 were significantly evolved. As shown in figure D, cluster 6 represents hepatic stellate cell populations, since many of the cells in this cluster express pluripotent genes like ACTA2 and CXCL14. As shown in figure E, the cells in cluster 7 represent populations of interzonal endocellular cells, in large part to the expression of the TNXB gene. In this slide, figure F shows that the cells in cluster 6 and 7 show the expression profile of several pluripotent genes, including COL1A1. In addition, the cells in cluster 12 represent portal-periportal endocellular cell populations, since most cells in this cluster express IFBP7 and SEMA6D genes. As for figure G, the cells in cluster 13 represent typical epicellular cells, while epicellular cell genes like KRT6A, KRT19, NTS, and S100A9 were significantly expressed. The next paper is from Journal of Hepatology, published in September 2021, named the Dopamine Receptor D2 Antagonization Normalized Pro-Fibrotic Macrophage Endocellular Crosstalk in Non-Alcoholic Steatohepatitis. Fibrosis in liver is the main determinant of non-alcoholic steatohepatitis, also called NASH, a disease that parallels the worldwide surge in metabolic syndromes. Currently, there is no effective treatment for liver fibrosis. Well, PPYAP signaling is essential for liver regeneration. Its aberrant activation frequently leads to fibrosis and tumorigenesis. Unraveling context-specific contributions of YAP in liver repair might help selectively bypass fibrosis and preserve the pro-regenerative YAP function in hepatic disease. The research strategy is shown below. At first, YAP level in macrophage increased in the liver with liver fibrosis. Deletion of YAP1 activates INF1 signaling and prevents liver fibrogenesis. And here, single-cell profiling was used to identify the changed cell populations. Then the researchers can get conclusion that YAP inhibitor DRD2 antagonist blocks fibrosis and restores hepatic architecture. The researchers have the results of single-cell sequencing analysis. Firstly, single-cell RNA sequencing analysis was performed on liver non-parenchymal cells, also called NPCs, to identify the 21 populations across 9 cell lineages. In addition, uniform manifold approximation and projection analysis revealed that the frequency of macrophage and EC populations were most significantly lower in the mice, which was verified by immunofluorescent staining. Finally, specific markers for macrophage like ADGRE1, CLEC4F, MACRO, and TREM2, and EC like PECAM1 and CDH5 were identified. and single-cell T-SNE analysis helped to reveal 5 subpopulations, among which the EC1 showed higher expression of profibrotic gene like CTGF and VCAM1. Okay, the third paper is from Science Translational Medicine. It also studied into liver fibrosis in non-alcoholic steatohepatitis. The paper was published in October 2021, named Targeted Epigenetically Maladapted Vesicular Niche Alleviates Liver Fibrosis in Non-Alcoholic Steatohepatitis. Non-alcoholic steatohepatitis, also called NASH, a rising cause of chronic disease paralleling a global increase in metabolic syndrome, leads to fibrosis, cirrhosis, and liver failure. Preclinical studies that systematically model the cellular and molecular networks underlying NASH pathogenesis will help in developing NASH therapy. However, the functional contribution of specialized liver EC subset to human liver cirrhosis or NASH pathogen has not been systematically sketched in complementary clinical and preclinical models at the single-cell level. The research strategy is shown below. At first, the research revealed the vesicular maladaptation and distortion of endocellular taxonomy in human cirrhotic liver. And then they constructed a mini-pig NASH model to find out the sequential targeting of DHAC2 and DNMT1 mitigates fibrosis. IGFBP7 and ADAMTS1 from maladapted SECs predict the progression and the therapeutic outcome of liver fibrosis in human and mini-pigs. And finally, they constructed a mouse NASH model to find out the roles of epigenetically maladapted SECs, IGFBP7, genetic inactivation of ADAMTS1 in a pro-fibrotic Th17. During this period, 10x single-cell sequencing was performed, and the researchers got conclusion as following. Figure A and B showed clustering 22. On 374 NPCs from 2 healthy and 2 cirrhotic livers revealed 25 populations. If we take figure B and C together, we can know 7 cell linkages were identified by expression of marker gene, including T cell, B cell, EC, macrophage, neutrophil, dendritic cells, and EPCAM+ cells. From figure D, we found that ECs exhibited the highest number of differentiated genes in the tested cell types, except for EPCAM+ cells with minimum cell numbers. Generally, the proportion of ECs increased in cirrhotic livers compared with healthy livers. Here are the results of the study. Overall, epigenetic-dependent hepatic vesicular maladapted enriches fibrogenic Th17 cells to promote liver fibrosis in mice, mini-pigs, and human patients with cirrhosis. Further analysis of human mini-pigs and mice suggested that crosstalk between histone deacetylase 2 and the DNA methyltransferase 1 promoted the liver EC maladaptation to promote production of IGFBP7 and ADAMTS1 in extracellular vesicles, recruiting fibrogenic Th17 cells to the liver. Pharmacologically, targeting of DHAC2 and DNMT1 alleviated the fibrosis in a mini pig NASH model. In summary, epigenetically reprogrammed vesicle adaptation contributes to liver fibrosis. Targeting of vesicle adaptation node might block maladaptive vascularization to promote liver regeneration in NASH. Here is the fourth paper from journal Hepatology, published in November 2021, named Group 2 Innate Lymphoid Cells Promote HCC Progression Through CXCL2 Neutrophil-Induced Immunosuppression. Due to their inherent characteristics, the function of group 2 innate lymphoid cells, also called the ILC2, varies in a context-dependent manner. ILC2s are involved in certain liver diseases. However, their involvement in HCC is unknown. In this study, it assessed the role of an HCC-derived ILC2 population in tumor progression. The scheme of the study is shown as the picture in the right. At the beginning, the researchers revealed the enrichment of ILC2 in the human HCC by single-cell RNA sequencing and FACS. And then they explored the liver ILC2's functions as immunosuppression in HCC. In addition, the researchers applied a mouse liver hepatitis model with single-cell RNA sequencing technology to reveal ILC2-induced immunosuppressive neutrophils through ILC33-mediated IL-13 expression. Finally, they constructed a mouse model to prove that liver KLRG1 ILC2 induced neutrophil immunosuppression, while depletion of neutrophil and the CXCL2 inhibited tumor progression. And here is the result of their study. Figure B is the 10x single-cell tsNE analysis. We can see certain gene clustering here. The researchers discovered that ILC2s were highly enriched in human HCC and correlated significantly with tumor recurrence and worse progression-free survival, as well as overall survival in patients. In the figure from the right, the researchers used mass cytometry and identified a subset of HCC-derived ILC2s that had lost the expression of killer cell lectin-like receptor, subfamily G number 1. It also called KLRG1, distinct from their circulating counterparts. These hepatic ILC2s highly expressed the CD69 and an array of tissue-resident related genes. So finally, according to their study, restoring KLRG1 in ILC2s, inhibiting CXCL2 in ILC2s, or depleting neutrophil inhibited tumor progression in a murine HCC model. So since we still have some time for live question part, let me take a look at the question in Q&A module. And I'll pick up 2 questions as the time is limited. So the first question is, what cell type can we accept for 10x single-cell gene expression library? We can accept the cell types that express poly-A mRNA modules with cell size smaller than 35 micrometer. Theoretically, the maximum cell size for 10x genomics is 30 micrometer. However, we had the successful experience with cell size a little bit over 30 micrometer. And here's another question. What species novel gene can accept for 10x single-cell RNA gene expression library? Novel gene 10x single-cell gene expression library is compatible with species ranging from human, mouse, rat, and other animals according to our sample requirement for single cells. But if clients send the tissues, the tissue dissociation technology is a limitation. We do not have a mature solution for some special tissues. So if you would like to send us tissues, so please feel free to contact your cells and your TS for further evaluation with our lab. Again, thank you so much for attending, and I wish you a nice day. This time I'll turn it back to Chloe.